The human heart, as I review in other videos, has a very specific structure. Two atria left and right, two ventricles left and right. The deoxygenated blood on the right half of the heart is completely separate from that of the oxygenated blood on the left side of the heart. The myocardium possesses cardiac muscle, which is only found in the heart. It is striated. It has a number of important characteristics such as the prolonged depolarization, which allows its contraction to be prolonged. There are junctions between cardiac muscle cells at intercalated discs, known as desmosomes and gap junctions. There is a specific pathway of blood flowing through the heart, so that the oxygenated blood is once again separated from the deoxygenated blood. The heart is lined by a serous membrane known as the pericardium, and this allows then the heart to be able to freely move because the pericardium has two layers, and in this pericardial cavity is a pericardial fluid which lubricates the heart. So, the human heart is a very complex structure, and its function is very important to us. But when we consider the vertebrates, the question leaps at us: where did hearts come from? And so that's what this lecture is going to focus on, and so that the history of those structures in the human heart is better appreciated. And so humans are not the only ones to have hearts. Large animals need some way to circulate body fluids with oxygen, with nutrients, with hormones to remove waste, etc. And then if fluid is going to transport these substances, it has to be pumped. And so that is the role of the heart to pump this fluid. And here you can see we have the hearts of a shark, an osteichthyon fish, etc. And as I will mention, as we study these hearts, you can see that the structure of the human heart and those of all vertebrates were developed gradually over long periods of time. So hearts did not begin with separate right and left halves of the heart, pumping blood to the lungs and back to get oxygen, or pumping blood to the tissues and back. So where do hearts come from? Well, it's complicated. And when we look at hearts, so for example, in a fruit fly do have blood vessels, and they do have a heart which pumps these blood vessels. The blood vessels, however, are what are called open, because in addition to fluid being inside blood vessels, fluid can then be just in the body cavity, the coelom. And if the blood vessels open into the coelom, and then this fluid, which is part, the blood and the coelomic fluid then comes back into the heart to be pumped again, then we don't like to call it blood per se, and rather a hemolymph, because it's not only retained within blood vessels. And so when we look at the structure and the evolution of hearts, invertebrates can have this open circulatory system, which then needs less pressure, because if the heart is not pumping a fluid which is always contained within blood vessels, if it then mixes with the body cavity, then when an insect moves, just muscle contraction will apply pressure which mixes the fluid. So the heart can be rather small when we deal with open circulatory systems. There are invertebrates which have closed circulatory systems as well. So here you can see the crayfish heart. Notice that, and I'll get to this in a second, where we vertebrates, we think of the heart belonging on the ventral side of the body and the nerve cord being on the back. But we actually see the opposite in most of the invertebrates, where the heart is on the back side of the body and the nerve cord is on the front. One of the early things that happened in the early deuterostomes is we feel that there was a reversal of the body so that the what was ancestrally dorsal and ventral switched, and so that when we compare hearts, we can look on opposite sides of the body. The dorsal hearts on the invertebrates being homologous to those of the ventral hearts on vertebrates. Now, when we say heart, you don't have to have just one. If a heart is essentially a contractile blood vessel, and if you want pressure, you can have more than one heart. Earthworms do. And so earthworms have a dorsal blood vessel, a ventral blood vessel, and connecting them are these contractile blood vessels, the hearts. Once again, hearts are typical, but then hearts can pump blood into vessels, which are then short and then mixed with the coelomic cavity. And so there are invertebrates which have hearts, which ask, the question, are these separate structures that evolve separately, or are they somehow homologous to the vertebrate hearts? Well, in recent years, the classification of the bilaterally symmetric animals has been reorganized. And while the new system is consistent with genetic evidence, it's a little more complex when it comes to the anatomical evidence, because there are hearts throughout this family tree, but then there are also now organisms which lack hearts. And so did hearts evolve separately? Did hearts evolve once, and some of the other organisms lose it? So in the previous classification, arthropods and vertebrates were coelomates, while nematodes were not. Annelids would also be, and mollusks would be coelomates, while some of these others like flatworms would not. And in that classification, it made sense. Oh, hearts evolved once in the ancestors of the coelomates. But now arthropods have hearts and nematodes don't. Annelids and mollusks have hearts, flatworms don't. So what is happening there? Well, you would want to suggest that maybe hearts evolved then separately. But the reality is that when the human embryo says, put a heart here, it's using a number of genetic signals that are the same signals as an arthropod embryo says, put a heart here. So the last common ancestor of these groups had to have had something in the way of a contractile blood vessel, because once again, the genes to make a contractile blood vessel are shared among different lineages. But once again, the reorganization of this classification scheme, it makes sense from the genetic sense. Anatomically now, a few things are less clear. When we look at the chordates, the invertebrates which are most closely related to the vertebrates, some have hearts. With the lancelets, we could argue as to whether the ventral blood vessel is homologous to a region of the heart, like the sinus venosus. By the time we get to the first fish in the Cambrian period and the most primitive fish alive today, there are actual hearts. So the hagfish heart is made of 3 chambers. A sinus venosus. We can use the word sinus for a wide chamber which has a thin wall, and here's collecting venous blood from different parts of the body, and is the sinus venosus, a chamber of the heart which empties into the one single atrium. The ventricle is single, and then this pumps the blood into the aorta, which goes from the heart. So the most primitive of the fish, the hagfish, and once again, anatomically, it seems hagfish are a little more primitive than lampreys. Genetically, it seems that they are about on par with each other. But hagfish are a bit more primitive in only having 3 chambers of the heart. They do have actual cardiac muscle. All right, so just like our hearts have cardiac muscle, so does that of all vertebrates. Previously, most of the muscle in hearts were made of smooth muscle, although, so for example, a hemichordate can have smooth muscle in its heart and a little striated muscle. Here it's all striated cardiac muscle. Unlike hemichordates, there is an epicardium around the heart. There is an endocardium lining the chambers of the heart. There are heart valves, which the invertebrates don't have, an atrioventricular valve, just like we have, a semilunar valve, and then also a sinoatrial valve between the sinus venosus and the atrium. Even the vertebrates which lose this then can possess it as embryos. In the chordates, while the heart is a contractile blood vessel, the blood sometimes goes one direction, sometimes it goes in the opposite direction. But now in all of the vertebrates, blood goes in one direction only through heart chambers, and obviously the valves help that stopping blood from going in the wrong direction. This sinus venosus is where a new heartbeat is initiated, and that will be true of all vertebrates. So even in me, even though the primary pacemaker is in the right atrium, it started off in my sinus venosus when I was an embryo. The sinus venosus smushes into the right atrium, and so now that primary pacemaker is called the SA node, the sinoatrial node, because it started in the sinus venosus. Some of the signaling involving the heart are the same, like epinephrine affecting the heart, neuropeptides affecting the heart making ANP, which is a peptide which affects water balance. Hagfish can control the output of the heart, so it's not constant. So the heart is not just a pump, it's an adjustable pump. So when we look at the hagfish heart, struck with a couple of things. One, so much more advanced than any of the invertebrate hearts, so much bigger, which is why hagfish can be large, can swim actively, but it's also primitive, as I'll mention in the next clip. There are no nerves which go directly to, or no major nerves which go directly to the heart. The heart doesn't have a blood supply. Right, and so this is muscle, but the fact that it's only getting deoxygenated blood, there are no coronary blood vessels, means that it can't work very well. So hagfish hearts clearly, more similar to those of the higher vertebrates than any of the invertebrates, but still the most primitive fish heart. When we look at lampreys, the other type of jawless fish alive today, we see the sinus venosus, the single atrium, a single ventricle, and then the fourth chamber of the heart, the truncus arteriosus, sometimes called the conus arteriosus. This is what will split to form, the aorta and the pulmonary trunk in human embryos. But when I was an embryo, I had all 4 of these chambers, just like the lamprey. So the lamprey, they have this conus region as well. The cardiac muscle has some of those junctions that I mentioned earlier, like desmosomes and intercalated discs, like that of higher vertebrates. Hagfish have a pericardial cavity. In fact, some of the invertebrates have a pericardial cavity, but in the invertebrates, the pericardial cavity doesn't completely surround the heart, and thus the pericardial fluid isn't, only limited to being around the heart. So there's communication with the coelomic cavity and coelomic fluid. So in an invertebrate, the fluid around the heart can be coelomic fluid. In hagfish, there's still a little of that. While there is a pericardial cavity, the coelomic fluid communicates with it, and so there is a mixing. That is also true of larval lamprey, that the pericardial cavity is connected to the coelomic cavity, but in adults, the pericardial cavity is no longer connected to the coelom, and that's like all of the higher vertebrates. There are chordae tendininae attaching to the AV valves. The vagus nerve reaches the heart, although it's just the sinus venosus. Nevertheless, that was not true of hagfish hearts. So here we have the autonomic nervous system reaching the heart, and the heart responds to acetylcholine. So that's in advance. The heart of hagfish doesn't. The interesting thing is that while it responds to acetylcholine, it doesn't respond as the hearts of the jawed vertebrates do. In lampreys, acetylcholine will stimulate the heart and speed it up, while in the jawed vertebrates, acetylcholine slows it down. So lampreys have features that are shared with the higher vertebrates that hagfish hagfish lack, but lampreys still have very primitive hearts. We get to now a shark. The heart is so much bigger. And sharks are now more active. Once again, we have these four chambers: a sinus venosus, a single atrium, a single ventricle, and then the truncus or conus arteriosus. And when we look at what sharks and all of the jawed vertebrates have, are coronary blood vessels. So the heart wouldn't be able to be very muscular and be very dependable if you couldn't get it oxygen, if there weren't coronary blood vessels, which are specifically giving oxygenated blood to the myocardium of the heart, then this muscle would fatigue rather easily. And so the coronary blood vessels of the hearts of the jawed vertebrates allow the hearts to get bigger, the jawed vertebrates to get bigger and be more active. The vagus nerve inhibits the heart. Acetylcholine and the muscarinic receptors inhibit the heart. So this is true of all of the jawed vertebrates, unlike the situation we saw in lampreys. Norepinephrine and epinephrine increase at both heart rate and contractibility, and there are faster impulses being spread in between the atria and the ventricles. And so the shark heart is more like that of the higher vertebrates than those of the jawless fish. When we get to the osteichthyons, the bony fish, we see the same heart chambers: a sinus venosus, a single atrium, a single ventricle, and the truncus or the conus arteriosus. It is more like those of the tetrapods. Epinephrine will dilate coronary arteries and constrict most of the systemic arteries, and the sympathetic nervous system of the spinal cord is helping to regulate the heart. That was something which we did not see in sharks. So before we get to the fish with lungs and the tetrapods, just a quick recap that fish have hearts, and we see many of the same structures. But if you were to look at the heart of a hagfish here, there's a sinus venosus, an atrium, a ventricle. These notice how small it is. Because it doesn't have coronary blood vessels to adequately bring oxygen to the cardiac muscle wall. The space around the heart, the pericardial cavity in hagfish, is connected to that coelomic cavity. It's not completely separated from it. Lampreys, once again, was a bit more advanced. So the pericardium completely surrounds the heart. There is a pericardial cavity. So lampreys have a heart. I'll be talking about the blood vessels and the flow of blood in the next video. But once again, when we get to sharks now, we start seeing larger hearts, more muscular. So here's obviously there's a pericardium And notice that the heart is now in a complete pericardial cavity separated from the body cavity, the coelom. There is an atrium. It looks like two atrium, but that's just one atrium that connects. There is a ventricle, and then here's the sinus venosus, which leads into the atrium, which leads into the ventricle, which leads into that conus or truncus arteriosus. There's that thin wall. See how thin the wall of the sinus venosus is? That's why we call it a sinus. And notice the coronary blood vessels. This muscle can get bigger because it has blood vessels servicing it, giving it oxygen, unlike the situation we saw in the jawless fish. We see much more muscle here. The ventricle has a much thicker, more muscular wall than the atrium does. That is like what we see later. There are valves, right? So there are atrioventricular valves and semilunar valves in these hearts, and then similar things in the hearts of bony fish like the gar and the bowfin that I have here. There's a sinus venosus, an atrium, a ventricle. There are valves. Now, once again, when we're looking at the history of hearts, this is about half of it. We don't get to tetrapods, until the end of the Devonian period. And so, a good 200 million years or more, if you go back to, the invertebrate hearts, a good 200 million years, hearts evolved in fish. Now, the amphibians evolved from sarcopterygian fish, like lungfish. And here there's something very important. Lungfish actually have lungs. And so there is a pulmonary vein coming from the lungs, but unlike most veins, this has oxygenated blood. Right, veins coming from muscles don't have oxygenated blood, but veins coming from the lungs do. So lungfish have lungs. So here's a vein that has oxygenated blood. So veins empty into here, bringing deoxygenated blood on the right half. Veins empty into this part, having oxygenated blood. So when we look at the sarcopterygian heart, while they have one single atrium, it is partially split. There is, the beginning of a separation between the left half, which is collecting the oxygenated blood, and the right half, which is connecting the deoxygenated blood. Amphibians will then split this and have two atria. So there is a partial interatrial septum showing a transition between other fish hearts and those of the amphibians. The sinus venosus empties deoxygenated blood into the right half. The pulmonary vein brings oxygenated blood into the left half, and the amount of cardiac muscle increases. Okay, so here you can see a lungfish and its heart. The sinus venosus, the atrium, the ventricle, the conus arteriosus. Okay. And so from sarcopterygian fish, then evolve the first tetrapods, the amphibians, by the end of the Devonian. So if you were to look at a frog heart, the great advance here is that now there are two atria. All right, so for hundreds of millions of years, there was one single atrium. Now there are two, a left atrium and a right atrium. The right atrium is getting deoxygenated blood from the sinus venosus. The left atrium is getting oxygenated blood from the lungs. There is still only one ventricle, though. All right, and so one could say, oh, that's horribly inefficient. The blood is now mixing. You're mixing oxygenated and deoxygenated blood. That could be true. However, it's more complicated than that, because both of these chambers empty into the ventricle doesn't mean they have to be emptying in at the same time. And so there can be other ways of keeping blood separate. Even if it's entering into one chamber, you can channel it so most of this blood is going over here or most of that blood is going over there. But this is obviously not as efficient as it would be if there were two separate ventricles, which completely isolated the two halves of the heart. And so the completely separate right and left atria, that is the major advance of the amphibian hearts. So if we were then to look at amphibians today in dissection, looking at a frog, there's a pericardial cavity. You can see that wrapping the heart. So here's the pericardial cavity. If you tear that, then you can see the heart. There's the large ventricle, but now we have a right atrium, a left atrium, a single ventricle, both of these atria empty into the single ventricle, and then the truncus arteriosus. And there is still a sinus venosus. So here's the large ventricle. I've cut the pericardium, right and left atria there. And then the same would be true of the salamander. So you have the ventricle, the truncus arteriosus, and two separate atria. This is about the same situation in reptile hearts. And so I'll get to that video next, but you can look at a turtle heart, once again, the very large ventricle, the truncus arteriosus, and then two atria right and left. And there's still a sinus venosus, although that is getting smaller. And so here you can see the heart of an alligator, the heart of a bird. Birds have completely separated. There is a complete interventricular septum in birds, but it's different from that in mammals. So birds have a four-chambered heart like mammals have a four-chambered heart. If we look at just more general reptiles, there is a completely separate pulmonary circuit, and this will be a topic in the next video. There's no ductus arteriosus, which allows blood to mix in adults. And once again, that'll be part of the next video. The sinus venosus is getting smaller. The conus arteriosus, or the truncus, may be present in the embryo only, and then makes up portions of the vessels which are leaving the heart. And a pulmonary, the pulmonary trunk and at least one systemic aorta leave the heart. There can be two systemic aortas. So if you ask what happens to the truncus arteriosus, that then becomes divided blood vessels, which are now separating blue deoxygenated blood and red oxygenated blood. These larger hearts generate greater blood pressure, they have a greater cardiac output, and there is an intrinsic conduction system, so that instead of muscles contracting in a wave going from one to the other, that there is these autorhythmic fibers which then carry the impulses that a new heartbeat has begun throughout the heart. So obviously the first reptiles date from the early Carboniferous, and then the reptiles have branched greatly, throughout time. So, when we look at the heart of a lizard or a snake or a bird or an alligator, it doesn't have to be, holding on to that early Carboniferous form. Evolution has continued in all of these lineages. Birds have such high oxygen demands for flight that their hearts are really large. I mean, they're about 40% larger than a mammalian heart relative to size. Some birds have hearts larger than others, and so aquatic shorebirds, kingfisher swallows, and hummingbirds have the largest hearts. Heart rate can vary. So an ostrich may have a resting heart rate, of around 40 with a maximum of 175, but songbirds can be, a heart rate of 350 or 450 heartbeats per minute, a maximum of 1,000. And then hummingbirds can reach 1,200 heartbeats per minute. And so obviously the demands of flight are quite intense, and so the heart has adapted to meet these demands. But not all birds would face this equally, like that ostrich there compared to the songbirds. And so how is this structured? There is a complete interventricular septum. So there is still a sinus venosus that brings deoxygenated blood into the right atrium, into the right ventricle. It then goes in through the pulmonary trunks to the left and right pulmonary arteries, to the lungs, where it becomes red oxygenated blood. This then returns through pulmonary veins into the left atrium, into the left ventricle, and then out the aorta, which is only on the right side, unlike in mammals where it's on the left side. And that I'll talk about that later. So this looks very similar to the situation in mammals, but it should be stressed that this interventricular septum evolved separately. So the first mammals split the one ventricle into left and right halves, and then the ancestors of birds did as well. Like I said, the first mammals, before we talk about mammals as adults, if you were to look at the mammalian embryo, you see a sinus venosus, which is like fish hearts, but not like the adult mammalian hearts. You see a single atrium, a single ventricle, that truncus arteriosus. And so the embryonic mammalian heart, including that of humans, looks like that of the ancestral condition in fish. But then during embryonic development, then the sinus venosus is absorbed into the wall of the right atrium, which is why the primary pacemaker is called the SA node, the sinoatrial node. It started off in the sinus venosus and ended up in the right atrium when the sinus venosus was absorbed into it. The atria splits into left and right atria. The ventricle splits into the left and right ventricle. The conus arteriosus splits to make the aorta and the pulmonary trunk. And so everyone's going to mention that on the SA node. And so this is then the situation we see in the adult mammals. But once again, the embryonic mammals, they have traits which we see in more primitive hearts. There's mammals have an AV node and a foramen ovale, which will join the two atria during fetal development. Obviously, fetal mammals are developing inside the mother, and the lungs do not have oxygen in them until birth. And so blood is diverted away from the lungs until after birth. When we look at mammalian hearts, they're essentially then the same. Here in an opossum, you can still see a little bit of a sinus venosus, which in the placental mammals, you don't in adults. So there you saw the vena cava were emptying into, so here you see the inferior vena cava, there's two superior vena cava, they're emptying into a common region before going into the right atrium. Here's an opossum heart, and notice that there is a complete interventricular septum, which is separating the deoxygenated blood on the right side of the heart from the oxygenated blood on the left side of the heart. That truncus has now split to make a pulmonary trunk and an aorta. And so there's the right atrium gives blood to the right ventricle, to the lungs, to the left atrium, to the left ventricle. There's the aorta and the pulmonary trunk. And so I'm just going to quickly go through this because in another video, then I will go through the anatomy of the heart. And now all mammals, it's about the same, right? So the right atrium, where is it getting blood from? An inferior vena cava, one or two superior vena cava, you saw two in the opossum, and then there's blood coming from the heart itself through that coronary sinus. There are semilunar valves separating the ventricles from the vessels. There are atrioventricular valves separating the atria from the ventricles. The ventricles are split, and you can see even on the surface of the heart, there's a shallow groove, an interventricular sulcus where some of the coronary blood vessels run. There is a pericardium around the heart, so you can see that here in the opossum. And so these hearts are largely similar. There's the pericardium. One difference you can see here is the placental mammals will have one superior vena cava, but here you can see two in the opossum. So that is a difference. But once again, the structures of the human heart, and then here you'll see the cow heart, the sheep heart, the cat heart. They're all about the same. So here you can see the pericardium of the cat heart. You see the interventricular septum. Note that the left ventricle makes up the apex of the heart and has a thicker wall than the right ventricle does. The left ventricle is pumping blood throughout the body. The right ventricle is only pumping blood to the heart, to the lungs and back. And so, this is essentially, here's the right ventricle pumping blood into the pulmonary trunk to go to the lungs. But this is fairly standard. If we were to look at the anatomy of the cow heart, we would see, all of the same structures, which, we see in the human heart, etc. Here, those chordae tendinonae attaching to the AV valves, etc. And so, rather than repeat myself over and over again and just go through, here's all of the structures. I have a video that talks about, the human heart, which once again is essentially the same, which we'll talk about blood flow through it. But once again, once you understand the anatomy of the heart in one of these placental mammals, you could identify, the structures in any of the placental mammals because of how similar they are. If you we're trying to identify, parts of the beef heart, specifically. Once again, I'm going to end, this video, but I do have videos where I go through beef hearts and their components. Here's an introductory chamber here, introductory video. Then I have one video that's specific to the heart chambers, showing how, if you were taking a lab practical and you wanted to get the question right, how you would distinguish between, the right atrium, the right ventricle, etc. And then I have another video on the valves of the beef heart, and then finally on the vessels. But like I said, I'd like to do a comprehensive, video on the anatomy of the human heart, and these are essentially the same. And so the structures which we see in the human heart, they've evolved. Hearts have evolved going back from before there were vertebrates. There were simple invertebrate hearts. And as we go from the jawless fish to the jawed fish to the bony fish to the sarcopterygian fish, which start to separate oxygenated blood and deoxygenated blood with a partial interatrial septum, to the amphibians, which separate it with a left and right atria, to the early mammals, which separated the ventricles into left and right independently from birds, which did the same. We see that hearts, have evolved over time, but that the placental mammal heart really hasn't undergone major changes since then. So once you know the structures of one heart in placental mammals, you would be able then to identify the structures of any.